Right then, Year 12. This is going to be the final video on respiration. And we're going to look at anaerobic respiration in both animal cells and in yeast cells as well. Okay, so you should all be familiar with glycolysis. If you can't remember glycolysis, here is a quick recap. It happens in the cytoplasm, and we're going to convert glucose into 2 molecules of pyruvate. Remember, glucose is a 6-carbon compound, and then pyruvate is a 3-carbon compound, so obviously there's going to be 2 of them to make sure we have the right amount of carbons. Now, first thing that will happen is the glucose will be given a inorganic phosphate from a molecule of ATP. We call that substrate level phosphorylation, and you can see it happening there. That will convert the glucose into glucose phosphate. That glucose phosphate will quickly turn into fructose phosphate, and then that fructose phosphate will be given another inorganic phosphate, so substrate level phosphorylation, and it turns it into a very unstable 6-carbon compound called hexose biphosphate. It's so unstable it quickly splits into 2 molecules of triose phosphate, each one containing 3 carbons. The triose phosphate, thanks to an enzyme, will now lose a hydrogen and lose an inorganic phosphate. The hydrogen will be picked up by a coenzyme called NAD, and the inorganic phosphate will be picked up by a molecule of ADP. So we've just made a molecule of ATP and a reduced NAD. That intermediate molecule will then lose another phosphate, and that will be given to a molecule of ADP, and now it's finally turned into pyruvate. So we now have 2 molecules of pyruvate, and we want to know what happens there. You would normally know in aerobic respiration they get transferred, they get actively transferred into the mitochondria, but that requires oxygen, and there's no oxygen, or there's a very low amount of oxygen. So we want to know what happens at this stage. So let's have a look inside an animal cell. Now, that pyruvate will be given a hydrogen. It's given a hydrogen from the reduced NAD that was made in glycolysis. So it's a bit of a cycle, as you can imagine. So there we go. That pyruvate has now turned into lactate, and that oxidized coenzyme NAD is now going to go back to glycolysis, where it can help with the continuation of glycolysis. It's so important that glycolysis continues to happen because glycolysis yields a net gain of 2 ATP molecules. So we're getting a little bit of ATP, not a huge amount, but at least we're getting just about enough for the cell to probably continue. So that is what happens in an animal cell. What about a yeast cell? Well, in a yeast cell, something very similar happens. However, what happens to the pyruvate is the pyruvate is going to lose a molecule of carbon dioxide, and as a result of that, it gets turned into ethanol. What happens then is the ethanol is going to be given a hydrogen from a reduced NAD, and it turns it into ethanol. So there's a lovely summary for you. This happens in a yeast cell. So this is obviously outside of an animal cell. It will also happen in a plant cell as well. So your pyruvate loses a molecule of carbon loses the molecule of carbon dioxide, so decarboxylation. That turns it into ethanol, and then your ethanol is going to gain a hydrogen, and that's thanks to the reduced NAD that obviously was formed in glycolysis. And what you'll see here then is that coenzyme NAD will now go back to glycolysis to make sure that obviously it can continue to happen, and therefore you have a net gain of 2 ATPs taking place there. And then your end result is ethanol. So I'm hoping that has explained both anaerobic respiration in animal cells and outside animal cells in plant cells and yeast cells as well. Okay, good.